Let's get started here. So just in terms of an outline, I'm going to just give a brief introduction, then talk a little bit about a few vignettes from the last roughly 8 years of my career, centered on lead identification, lead optimization, and then if we get to it, some clinical insights that we've been able to pull out of some work that we've done more recently. So just in terms of kind of background, again, I'm part of the MD Anderson Institute for Applied Cancer Science, or IACS, which is part of the Therapeutics Discovery Division. So what we do is really focus on small molecule therapeutics. We've got another group within the division called Orbit that focuses on biologics. And then we work very closely with our two biology groups, Traction, which is really focused on innovation in kind of cancer biology, as well as a neurodegeneration consortium, which obviously is interested in neurodegeneration. Down at the bottom, I've got a fairly recent photo of the group. So we have about 100 scientists working at MD Anderson under this umbrella. And what's really pulled us together is this idea around having access and working within an environment that's different than pharma, where we have or can get insights into clinical research that's going on and use that to drive some questions and models and things in the biology space that then help to drive our drug discovery. And in that way, kind of bring things full cycle back to the patients in a more efficient manner. And just to give you an idea of the kind of what we've been up to over the last few years. Since we started in kind of late 2011, early 2012, we've been able to really push a number of projects forward. So I pulled this pipeline slide off of the MD Anderson website, and it really illustrates the fact that we've been able to move things kind of to the right of the pipeline and get some compounds into clinical trials. So at this point, we have 2 small molecule projects that have entered clinical trials, as well as 2 biologics. You can see down at the bottom that we have a number of other projects that are kind of close behind that. So the group here has really worked hard to make this kind of drug discovery within kind of an academic situation really kind of hum and deliver for patients. So I'm sure many of you have seen kind of this generalized drug discovery and development pipeline, starting all the way at the left side with target ID and on the right side with market. And each of these chevrons along this path aren't really the same size. As you get to the right side, the costs and the length of time really does kind of grow. But when we start talking about drug discovery and how computational chemistry can impact the process, we're thinking more about the sections to the left. There are some specialized tools that get used later on in clinical development, but we're really focusing in on things that are a little bit earlier on. And some of the things that come up and that you may have heard about are being listed on the screen. And I'm not going to get into detail on all of these, but what I'm going to do is just talk about a few vignettes around some major topics like virtual screening, using structure to guide design, and then potentially some molecular dynamics. And that's going to touch on some of the other circles in this illustration. So kind of moving back to the hit ID stage, oftentimes we're thinking about virtual screening, and thanks, Matt, for really laying out a kind of a nice framework for what that means and the way that those of us that are in the trenches really think about virtual screening. So what I'm going to do is walk through a case study of a project that we worked on when I was at Cubist, and this started in roughly the 2011-2012 timeframe. So we were interested in a GPCR target, CCR9. And what got us interested in this was that chemokines and the receptors are important in immune cells, and they have a lot of different functions in terms of things like viral entry, tumor growth, metastasis. The CCR9 for us was really interesting in that it's responsible for leukocyte recruitment in the gut and potentially for inflammatory bowel disease, which is something that afflicts a large number of people. What got us really interested in kind of following along is that there was an antagonist that made it to phase 3 clinical trials, but this failed due to limited efficacy. And when we took a look at the data, it looked like they weren't really able to dose the compound up high enough, that they had problems with the molecule itself. It wasn't necessarily the target, but likely the molecule. So we figured if we could come in and find some other chemical matter that was better behaved, we might be able to move that towards the development. So what I'm showing on the right is just a kind of the generalized homology model that we had put together for CCR9 with the tool compound that had been in the clinic, bound in kind of the extracellular orthoscopic site. So when we were thinking about virtual screening, we really wanted to hit this one hard, use essentially all the information that we had available to us at the time. So that included things like curated databases. At the time, the GVK, GoStar was one of the most extensive commercial databases available, but we also used things like Campbell and PubChem. We used the scientific and patent literature around the clinical compound, as well as our structural model. So really trying to cover our bases here. And then hit this with as many different kind of traditional virtual screening methods as we could. So things like at the top, a number of different cheminformatics type methods like 2D similarity searching, trying to identify privileged chemokine receptor structures, as well as searching for pan-chemokine compounds. We did a lot of this with the KNIME platform. Then moving down, we used 3D shape methods. We used rocks in this case from OpenEye, based on the Vercernon structure. And we also used pharmacophore-based searching methods that are implemented within Mo. And then for docking, we used the Gold software from CCDC. So we had a lot of inputs into kind of the compounds that were being selected by all these different methods. And the way that we did the data fusion really, I believe, influenced the way that the results that we got out at the end. So there were data fusion points kind of for each method, and I'll get into at least one of those in a minute. But once we had selected compounds out for each of these methods, rather than using kind of a traditional consensus scoring method where we wanted to see compounds that hit a lot of these different methods, we used really just a parallel selection. Rather than doing that, we selected everything and just pushed it all forward. Again, I think maybe something, going back to something that Matt had mentioned, not every compound that you're going to find is going to be found by all of your methods. So you may find a really interesting hit that only shows up in docking. Or you may find a really interesting hit that only shows up in 3D shape. And you don't want to throw that out because it didn't show up in your 2D similarity search. So again, I'm going to walk through what we did in the kind of looking for privileged structures just as an example of kind of this earlier stage data fusion that we were doing. And so if you start up on the top left corner, essentially we took all of the known CCR9 actives and fragmented them. And then up on the right-hand side, we looked at the public chemokine actives from places like PubChem and the GoStar database, fragmented those. But we also wanted to try to throw out just kind of standard medchem-like structures and really focus on the pieces that looked like they were more chemokine-centric. And so we compared those against a public kinase database and tried to figure out kind of a likelihood score that each of the fragments was really kind of chemokine-centric rather than just drug-like. And so to do that, we used things like normalized frequency based selection. We used adjusted frequency based on complexity and flexibility, as well as kind of scoring for matched pairs where you're really looking for compounds with similar structures but dissimilar frequency in them showing up in the chemokine versus the kinase set. And so once we had that, we could we could then move on and put together our virtual library, kind of down at the kind of the bottom left. Then use that to substructure search against a large commercial database of purchasable compounds to try and find actual molecules that we could get our hands on. And then went through and did some manual inspection and really saw whether we could purchase them, and then kind of fed that into the larger virtual screening process that I showed you on the previous slide. So at the end of the day, when we purchased in compounds, we ended up with just over 1,000 compounds that we purchased and screened at 20 micromolar. We ended up with about 200 hits based on an inhibition of greater than 30%. We ended up with about 60 hits with an inhibition above 50%. And later on in the project, we actually, during this process, we had initiated an HTS. So later on in the project, the HTS actually read out. And so comparing our results to the HTS, it looked like we were comparing pretty favorably. So we were enriching what we were bringing in. Now, if we break that down by virtual screening method, which is what we're looking at here, it's no surprise that the pan-chemokine compounds, which we already knew hit a chemokine receptor, had a high hit rate. Same thing with 2D similarity, since you're really searching kind of very close to known actives. Now, when we took a look at the chemical space coverage compared to CCR9 antagonists that are shown in red, again, what we see is that some of these methods, like the 2D similarity and some of the privileged structures, are really covering a lot of the same chemical space, but some of the others are really kind of exploring a little bit more. So things like the pharmacophore-based methods and the docking-based methods, you can see that they're covering kind of that lower right quadrant of the PCA plot a little bit more effectively. And so what I'm going to do next is just walk you through kind of quickly some of the hits that we found, and starting with the privileged structures. So on the left-hand side, you can see one of the analogs of the clinical compound, about 430 nanomolar. And we had a really hard time in our medicinal chemistry operations trying to replace that sulfonamide. Sulfonamide's kind of a special group in terms of the geometry that it can adopt. And so we had a really hard time. Now, the analog that you see to the right was something that was found in the privileged structure search. And you can see that this kind of extended a region between aryl groups with the acid sitting kind of in the middle only loses you about 20-fold in activity. And so this was really the first case that we had seen where we could at least start to think that we might be able to modify that mid part of the compound. What we found was that carboxylic acid was really critical for activity, and that we could replace and use some of the SAR from the original clinical compound on that scaffold as well. Now, we didn't go very far into the chemistry on any of these series, so it's really just early SAR that I'm going to be showing you here. For the pan-chemokine compounds, we were able to find a pretty interesting pyrazolopyrimidine hit. It was about 11 micromolar. And in this case, we found that the homopiperazine at the bottom of the compound was something that we could replace and modify with either linear chains or piperidines. And then moving on to compounds that came out of the pharmacophore search. Again, we had a very different type of chemotype here with a pyrazole core that we could include other substitutions on. But what we found here is that we really couldn't get away from that phenol up on the upper right. So I'll just take a minute to discuss some of the docking. So remember that we had our docking model based on the homology model that we had produced of Vercernon in CCR9. And we were able to find some really novel hits using that. So these compounds that I'm showing here, we had very nice docking models of those compounds making very specific interactions. Early SAR made a lot of sense. And again, remember that the compounds were based on the model of the Vercernon kind of sitting on the extracellular part of the GPCR. Interestingly enough, a few years later, the X-ray structure of this compound bound to CCR9 was released, and it's binding on the inside of the GPCR on the intracellular surface. So even though we had designed a homology model and refined this homology model against a kind of a faulty binding mode, we still were able to find things. But that could also be the reason that our hit rate was lower than expected with the docking. So this project actually was paused at this point and kind of shelved by the company due to a strategic business decision. Not due to science, but due to a refocusing of the company in a different direction. But we've used a very similar strategy at IAX in a project where we're looking for allosteric kinase inhibitors. And at the time, we were able to prosecute a virtual screen using shape, pharmacophore, and fingerprints against a library of about 11 million commercial compounds using a very similar data fusion type method, really looking at diversity as well as commercial availability. We were able to purchase in about 300 compounds, get a hit rate of about 11% on those, and after kind of confirmation, dose response, and triage by chemistry, ended up with about 7 hits that were prioritized for follow-up. And so from those, we've also been able to get X-ray structures where, in this case, you can see the kind of blue mesh blob is the electron density for one of the compounds. It's not engaging the hinge, so we were able to identify novel compounds that are binding it in the region of interest. So I'm going to change topics here a little bit and move on to some of the problems that we've faced in lead optimization. Now, I'm not going to talk about everything that we've faced in lead optimization, but really focus in on a couple of issues. So SHIP2 was really a project where we're relying on scaffold hopping in order for us to kind of move forward and identify proprietary tool molecules. The reason that we're interested in SHIP2, being a cancer center, is that you can see that the protein kind of sits in between receptor tyrosine kinases and the MAPK pathway, and you see our friend RAS kind of sitting there in the middle. And so it's involved in things like in a variety of cell functions like proliferation, differentiation, transcription. The protein itself has 3 domains, and you can see on the right, there's a catalytic site on the phosphatase domain, and this is actually covered up by one of the 2 SH2 domains. Basically, it's auto-inhibited. Now, when it's in the presence of these phosphoproteins, like you see on the intracellular side of receptor tyrosine kinases, the SH2 groups bind those phosphotyrosines, and then the catalytic site is opened up, and it can do what it needs to do to activate the rest of the pathway. So we've been looking at the target for a while, and back a few years ago, there was a literature report that claimed that there were compounds that inhibited SHIP2 by interacting with glutamate 250. And when you take a look at where glutamate 250 is, it's not in the catalytic site. So it's elsewhere. It's kind of at the interface of the 3 domains, or the 3 domains in SHIP2. So we went about trying to put together a binding model. So we used GLIDE to dock to the phosphatase domain and found what we thought was a reasonable binding hypothesis. So we used that as input to try to design new cores. So we used kind of medchem intuition. We used the recore method and the Mo scaffold replacement. Essentially what those do, if you look up in the right, is you can kind of erase part of your molecule and using kind of geometric constraints, try to fill that in. And we tried to fill that in using a combination of Cambridge structural database linker libraries, so information from small molecule crystal structures, as well as excluded volume from our structural model. And so what that looked like was essentially using a template and then coming up with a bunch of different ideas. And we needed to kind of rank order these and see if any of them looked like they might be able to move forward. So we used a combination of GLIDE, Mo, and a local strain estimate to do that. And what we found was fairly quickly we could jump to this pyrazolopyrimidine lead, IAX 13646, which had good enzymatic activity, good activity in cell. And we were able to get a crystal structure of this to see really what was going on. And so when we took a look at the structure had actually all three domains, not just the phosphatase domain. So our overall binding mode that we had gotten from docking was correct in that we were making the interaction with glutamate 250 in the right way. But because we didn't have the full protein in our model, we were missing out on some of the other interactions. So things like the interaction hydrogen bonding and pi cation stacking with arginine 111 on the right side, and some additional hydrogen bonding on the left side with the basic amine tail. What this did is it let us really help medicinal chemistry drive the idea creation process. We had relatively predictive models in terms of things that were going to fit and things that were going to be fairly potent. Unfortunately, the series suffered from transporter-mediated elimination issues, and after making a fairly large number of compounds, we couldn't improve that, and we deprioritized the series. However, since we had already been through the scaffold hopping exercise, we could go back to that and very quickly found a different core, pyrazolopyrazine. And here, the idea was really to look for cores that had fewer H-bond donors, since this is often a recognition element for transporters. We could immediately translate all of the SAR. All the interactions were maintained, as shown down on the right. And we ended up with a very efficacious compound that was selective across the phosphatases. Because there were some donor acceptor pairs in the compound, we ran that against a kinase panel at DiscoverX, and it was clean there. And we ended up with a good tool compound that had good PK properties and good bioavailability. Now we were also able to show that it was on target, and part of what we did here was we looked at not just SHP2, but also structures of SHP1. With the idea that we wanted to try to prevent compound binding and really show that our compound was hitting the target that we thought it was. And so we identified a residue in SHP2, this proline, that in SHP1 is a glutamine. And if we were to kind of in silico mutate that, it suggested that we should be able to keep the compound from binding and retain the overall structure of the protein. And that's exactly what we saw. So when we tested the SHP2 inhibitor, we saw the sensitivity change. When we used a MEK inhibitor, which is downstream from SHP2, we saw no effect. So that suggested that we were on target. The compound also has activity in RTK altered cancer and KRAS mutated cancer. So what I'm showing here is just the anti-tumor effect that we're seeing in a KRAS G12C model, where when there's no treatment, you can see the tumor volume increasing over time, and treated with 13909, you're seeing tumor stasis. So just going to switch gears for a few minutes here and talk about another one of our kinase projects that we had run at IACS. And this is around CSF1R. And the issue with this project was really that we wanted to improve selectivity. So again, depending on the problem that you're facing, you may use different techniques. Now, the reason we're interested in this is that it's a potential target for cancer immunotherapy due to CSF and CSF1R having an effect on macrophages and tumor-associated macrophages having an effect on the tumor microenvironment. There was also information available at the time that treatment with the CSF1R inhibitor correlated with the tumor-associated macrophage presence and attenuated tumor growth. So we didn't have structural biology capabilities on this project, so this was really driven primarily through structure-based means and docking. So what we did to start out with was modify the hinge on a tool compound that we had pulled out of the literature. So again, we wanted to change this out, see if we could get some additional selectivity. So essentially we wanted to swap hinge binders. And so we did that a couple of different ways. We used things like ReCore and the Mohs scaffold replacement that I introduced before. In this case, instead of removing the central portion of the molecule, we're looking to remove the hinge binder, find things that fit geometrically and could make the hinge interaction. We also used the breed method within Mo and using some of the aligned kinase databases that are provided there, where essentially what you're looking to do is generate hybrid molecules between something that might be binding to a different kinase and the rest of our core. So once we had went through that process, we generated something on the order of a few hundred potential compounds. We docked those and evaluated them, and we ended up with an aminopyridine compound that was actually had very good affinity. And what I'm showing you here is just the docking model that we had used to really drive a lot of the structure-based design after that. So unfortunately, our tool compound ended up having relatively good selectivity across the kinome, but really poor selectivity against very closely related kinases. And that's illustrated here by the DiscoverX kinase panel, as well as kind of taking a look at the KDs that we had eventually gotten for that family. So while there might have been more than 100-fold selectivity for some of them, in some cases you're only 20 or 30-fold different. That's really not what you're looking for in a kinase program. So what we did is we went ahead and took a look at the sequences of that family, and we identified a position that was unique to CSF1R relative to the rest of the family. So there is a glycine in CSF1R that's a cysteine in the rest. And when we took a look at where that sat, it sits right in, essentially in our binding site. So where we see a small pocket at kind of the south end of the core in CSF1R, that's being filled by cysteine and proteins like c-Kit. So the strategy here was to both explore that southern area as well as kind of a northern area where it looked like there was a little bit of extra space based on some of the X-ray structures. And where that took us is we were able to significantly improve the selectivity profile, both by going to the northern end, where you can see that we've gotten everything now above kind of 1,000 or above 100-fold. But what really was sort of shocking was how well the southern end worked. So we really didn't end up losing anything in terms of affinity for CSF1R, but at least in terms of just your binding to the other kinases in the family, we essentially abolished all of the activity in those with a fairly simple modification to the core. Now, at this point, I'm going to skip over a whole lot of really elegant medicinal chemistry work that got us to the eventual tool compound and just show you where we landed. So with IACS 9439, we were able to keep the KD for CSF1R at 1 nanomolar and have at least 450-fold selectivity against the rest of the family. which was really where we wanted to be. We ended up with a compound that was potent in cell, had good bioavailability, so we could dose it and show that it was efficacious in cancer models. And if you'll indulge me for just a few more minutes, I'd like to talk about something a little bit different here, where we're able to gain some clinical insight by taking a look at and working with one of our clinicians at MD Anderson. So this is work in collaboration with the HAMAC lab. And they're interested in tyrosine kinases, receptor tyrosine kinases, and they've been looking at things like HER2 and EGFR in a lot of detail. In particular, taking a look at the types of mutations that happen. So HER2's amplification of HER2 occurs in a lot of different cancer types, and targeted therapy seems to work in those cases, but there are also a lot of activating mutations, and you can see those along the bottom here. So those occur in both the extracellular domain as well as in the intracellular kinase domain. And there aren't any approved therapies for these. Now, what the HAMAC lab has done is they've kind of, in some of their cellular models, shown that a lot of these activating mutations show differential sensitivity to tyrosine kinase inhibitors. And when we took a look at where a lot of these mutations sit, it's not obvious why that might be. So most of these mutations are not adjacent to the inhibitor binding site. So you can see the inhibitor in cyan kind of hidden back behind the beta sheet. And some of the key mutations are sitting really out, not even on the periphery of the binding site, but not really interacting with the binding site at all. So when we took a look at this, we really couldn't explain this via something like docking. Essentially, we could dock all of the compounds into the binding site very effectively, given the X-ray structures that are available. And even using, we didn't use FEP, but we were using things like MMGBSA. We really couldn't distinguish one compound from another to any great degree, and certainly not be able to rank them like you can see in the experimental data that the HAMAC lab had put together. So essentially what you're looking at here is a series of mutations along the left-hand side and a series of tyrosine kinase inhibitors along the bottom. And so this heat map gives you kind of an idea of the sensitivity of each of these compounds, or each of these mutations for different compounds. So what they found was that poziotinib seemed to work across a lot of these mutations. And when we started to take a look at poziotinib versus some of the other structures, say ibrutinib, what kind of stood out was that poziotinib had a smaller kind of right-hand side. And when you line those up and kind of visualize where the alpha-C helix is in relation to the rest of the molecule, it looks like some of the molecules that had kind of poorer profiles ended up being larger and kind of sticking their tails out closer to the alpha-C helix. So even though all of these compounds appear to be able to bind to HER2, it seemed like a reasonable hypothesis that maybe size is responsible for some of this differential sensitivity. And so that's exactly what we tried to, we started to try to take a look at by taking a look at a couple of pairs of these mutations. So this L755 mutation, both to serine and to proline at the same position, and then the V777L and this duplication at Y772, which are kind of along the alpha-C helix. So in order to do that, again, docking wasn't going to work. So we figured that maybe there's something happening to the dynamics and the motion and kind of the conformations that are being sampled by the protein itself. So in order to do that, you might need to run very, very long dynamic simulations in order to capture these kinds of conformational changes, something on the order of microseconds to milliseconds. And we're not prepared to do that. We're not really capable of doing that the way that we're set up at IACS. We can do things on the nanosecond time scale, however, and so we used accelerated MD as a method of choice here to try to expand the sampling. And the way that it does this is to raise the minima of your potential energy landscape that's below a given threshold. Essentially what this does is it reduces the size of the barriers between minima and allows you to sample more space more effectively. So we used the implementation in NAMD to do this. And the results that we saw were pretty interesting. I mean, we were able to, in the case of the V777L versus the duplication mutation, see a difference in sampling between the two, in that the V777L really preferred to be in the kind of the alpha C helix out position. So that's seen on the right-hand side with the blue lines kind of being above the dotted lines in the center there. And the duplication mutation kind of sampling both states, so kind of the in state and the out state. And the overlay that you see on the bottom left kind of illustrates exactly how far apart those helix positions can end up being. So there's a fairly significant difference in the amount of space in the binding site in those different MD snapshots. Looking at mutations at the same position, we actually saw some pretty major structural changes as well. So with the L777 or L755P mutation, because that's sitting, that residue is sitting between two of the beta strands in the beta sheet in kind of the northern lobe of the kinase, this breaks a hydrogen bond between the beta 3 and the beta 5 strands, and that destabilizes the entire sheet. And that seems to have a knock-on effect where during at least some of the simulation, you're rearranging and kind of destabling the kinase hinge. So it's adopting some different conformations than you might expect just taking a look at a lot of X-ray structures. We don't see this, however, with the L755S mutation, which was a lot more similar to wild type. And again, this is consistent with the differential sensitivity that we had seen between these two mutations. So in summary here, really what we found was that the binding pocket dynamics and size appeared to play a key role in the tyrosine kinase inhibitor sensitivity for the HER2 activating mutations, and that some of the smaller TKIs like poziotinib, those can fit into most of the protein conformations that a lot of these mutations are sampling. Whereas some of the larger TKIs like ibrutinib, they can only fit into a kind of a smaller population of these. And so this may explain what the lab's actually seeing in terms of the differential sensitivity. And again, this is something that if we had just relied on docking, we wouldn't have been able to try to sort this out or even put this forward as a hypothesis. So just to kind of quickly summarize, I hope that I've been able to show kind of a few different methods that we've been able to apply across the drug discovery pipeline. Using multiple methods, and this is just my opinion and my experience, but using multiple methods in lead identification and virtual screening and leveraging all the available information really increases your likelihood of success. Thinking about data fusion is critical here. I'd recommend, once you're bringing information together from multiple methods, to avoid a kind of a strict consensus and really focus on parallel selection of your best hits from each of your different methods. We touched on a couple of different examples in lead optimization of things that you can do to try to improve the chemical matter that you have. But really, depending on the problem that you're trying to solve, you're going to want to tailor your approach appropriately. And then finally, just that we've been able to, I think, pull some useful insights out of clinical data, and in this way really suggest new ways that we might be able to approach inhibiting well-studied drug targets. So in the case of HER2, now we've got new design ideas and design hypotheses that we might be able to use to drive a new, kind of a new generation of TKIs. And I don't have a kind of acknowledgement slide because it would run to several pages. But I'd just like to call out Vijayan Ramaswamy. He's done a lot of the work, both in terms of the allosteric kinase inhibitor virtual screening, as well as the MD simulations that we've shown at the end here.